Montgomery Botanical Center is a place for research. Botanic gardens contribute a lot of very interesting research findings to the scientific community. But the challenge for a botanic garden is to figure out a way that they can make a significant contribution to science given the set of resources that botanic gardens have, which in many cases is not the same as what's available at a university. What our organization decided a long time ago was the absolute best thing that we could do was to keep doing what we're very good at, and that is growing an exceptional collection of plants. And then in order to get research done, to set up very good, productive collaborations with research experts, which often come from universities, but they may come from other kinds of places like museums or other botanic gardens. So really, again, it starts with an exceptional plant collection, and we get things done through these really productive collaborations.
I have a research associate appointment at the Montgomery Botanical Center, which allows me to pursue my research on the morphology and anatomy of plants with specific emphasis on tropical plants. My affiliation here is promoted by the fact that the Montgomery Foundation has emphasis on two major groups of tropical plants: the palms, on which I've worked for many years, but more recently on cycads. And in pursuit of these research activities, I've produced a number of books, most recently one on palms. Incidentally, of course, using material from various sources, but to a large extent the Montgomery's collections. The more recent research relates to the cycads, this very ancient group of seed plants, greatly facilitated by the fact that the collections here have represent something like 70% of all cycad species.
So it's very interesting that there are so many things that we can learn from cycads, and that's what makes the living collection so important here. But not just for the ones that have survived for so long, but we can actually learn quite a bit about some of the ancient cycads that we only know from fossils by looking at what we have now.
I'm interested in the fossil history and evolution of cycads. Present-day modern cycads capture attention mainly due to their ancient habit. But I think fossil cycads raise many more questions still waiting to be answered. So I'm engaged in the study of fossil cycads, and for this I need a comparison with the modern ones. This comparison includes not only the gross morphological study of the leaves, cones and seeds of cycads, but also their micromorphology and anatomy. For instance, the anatomical study of the epidermis and cuticle is the outer covering and protecting layer of leaves, which is often preserved in the fossil leaves, help to reveal the systematic relation of between the fossil and modern cycads.
So we have a lot of visitors that come here for structural biology, that is morphology and anatomical studies. People from all over the world come to visit here because we have such a nice plant collection. We're also very excited to be able to offer these collections for research by students to do very innovative projects in that area.
My research here at the Montgomery Botanical Center is focused around the genus Dione. I'm doing a complete sampling of the leaf cuticle micromorphology, looking at the internal structure of the cuticle under a scanning electron microscope. The idea of this is to build a quantitative database from qualitative characters to then use in statistical analysis. This work is in collaboration with Dr. Andrew Volvides in Jalapa in Mexico.
Well, my involvement with the Montgomery Foundation was the Montgomery Foundation. Then was during a visit, Knut Norstock came to the Botanic Garden to see the cycad collection, was very impressed, and arranged for me to have a postdoc here with Montgomery funding. And eventually we signed an MOU with Fairchild Tropical Garden when John Popenoe was director. We have a sister garden relationship with us. And I've been on and off giving talks and courses at Fairchild and Montgomery over the years.
So we've heard a lot from our visitors that are here to work with the cycad collection. But let's not forget we have a wonderful palm collection as well here at Montgomery. And that's a good opportunity for us to talk about one of the best ways that we can get research done here at Montgomery. Going out into the field and searching for these plants in order to build the living collection is actually a research-based process that has a lot of very interesting scientific outcomes.
Currently, my research involves the genus Cyagrus, which is the sister group to the coconut. Right behind us, we have a queen palm, which belongs to this genus. When I first started studying this group, there were between 30 and 31 species. Now we're up to 51 and still counting. I'm going to be going in January this coming year to Brazil again to do one last collecting trip. And I'll probably be there for about a month or a month and a half doing that. And we probably will be discovering some even more species, and I hope to make that final trip and terminate and finish up this group that I've been working on for probably about the last 20 years. So that's basically what I'm focused on right now is the genus Cyagrus.
So that tradition of research is something that's been going on for a very long time here at Montgomery. All the way back to the founder, Colonel Robert Montgomery, who established the plant collection, and the founder of the Botanic Garden, Nell Montgomery. We've been working to support scientists with unique plant collections and a place where they can work with them.
So I first came here in 1975 as a postdoc with Barry Tomlinson when he was at Harvard, but I was stationed down here. And half of my time was on monocots, and the other half was whatever I wanted to do. And I'd always liked cycads, and there was this huge cycad collection here that nobody was using or doing anything worth besides just kind of watching them grow, which really isn't very exciting. So and then when I tried to work on their anatomy and development, the sort of first conundrum was the taxonomy was really bad and nobody really could identify anything or know what it was. So I had to sort of tackle the taxonomy and the structural botany at the same time. So that just sort of started my whole career in cycads. And this collection has been fundamental to this, and for several reasons. And it's grown in value through time as we realize with these endangered species that we can utilize them to understand what's going on in habitat. Now, this does ex situ conservation, which isn't the same as being in habitat in a way, but what it does is allow us to study the plants and their biology so that we can understand something about their biology. We can then correlate that with things that are going on in habitat and understand the mechanisms by which they have survived and changed through time. Just because these are ancient plants doesn't mean they haven't changed somehow in the past, whatever it is, 250 million years. That is, a lot of the changes are very subtle. They may be changes in the genome that do things in their physiology that allow them to adapt to dry habitats, for example. So if we can understand the features, structural features that have happened in correlation with that, we can now take these plants and go back and understand the genes that underlie those changes. And then we can utilize that genetic information and actually apply what these plants have done in solving their environmental problems to problems that our crop plants might have that we'd like to exploit.